Good afternoon, everyone. My name is Justin Thiel, and I'm joined here today by my classmates Jay and Bryanna. The topic that we have chosen is color blindness in humans, specifically pertaining to red-green color blindness. As I previously mentioned, the subject of our research pertains mostly to red-green color blindness. There are also variants of blue-yellow color blindness, as well as complete color blindness, or monochromacy, and that's when an individual cannot see colors at all, but it's extremely uncommon. So within red-green color blindness, there are 4 subtypes. The most common among them is deuteranomaly, and it is a condition that makes green look more red. It's very mild and does not usually affect day-to-day activities. Second is protanomaly, which makes red colors appear greener. Next is protanopia, which makes individuals unable to perceive any red light, and deuteranopia, which makes someone unable to perceive green light. Anyone with these last 2 conditions would never be able to even tell the difference between red or green because they simply can't see one, so they have nothing to compare it to. Their mind would not even be able to comprehend what either green or red looks like. Color blindness in general is an X-linked recessive inheritance trait. Because of this, it is much more common in males than it is in females. The reason for this is that, as we know, females have 2 X chromosomes, while males only have 1. This means that females must have 2 copies of the recessive gene on each chromosome, whereas males only need it to be on the 1 X chromosome. This is why females have a significantly lower chance of being colorblind, because it has to be passed down by both parents. Additionally, this means that there is a 50% chance that daughters carry the gene and pass it on to the next generation, or they may not carry the gene at all. The picture at the bottom helps depict a couple of scenarios, both in which only one parent carries the gene. There is a 50% chance that sons will not carry the gene and have perfectly healthy vision. However, this also means that they have a 50% chance of inheriting and expressing the trait disorder. To bridge the connection between Mendelian and molecular genetics pertaining to this type of color blindness, let's take a look at where the specific genes responsible for this disorder are found. We know that the X chromosome is responsible for passing this trait on, but more specifically, it is the OPN1LW and OPN1MW, which are the genes on the X chromosome responsible for this disorder. Here is a visual representation that shows us relatively where these specific genes are located. We can see that the genes responsible for color blindness are located near the telomere of this chromosome. These genes provide instructions for making a protein found in the retina that is essential for normal color vision. And now Jay is going to explain some aspects pertaining to the molecular side of things.
So going into more of the structural reasons behind how colorblind works, it starts with the cones and rods of the eye, which can be located in the retina in the back of the eye. These cones and rods determine your vision, with the rods adjusting for your low light levels, such as during the nighttime, and the cones determining the high light levels, such as during the daytime. The cones are the ones that actually affect color blindness, but the rods still contribute to your overall vision, so they're somewhat still important. Cones come into 3 different types: the L, the M, and the S cones. Each contains a gene that affects the pigmentation of the opsin, making them more sensitive to light. So the image on the right shows the location of the rods and cones on the eyes, while the image to the left shows the sensitivity of the L and the M and the S cones. For right now, knowing the sensitivity isn't crucial for the cones, but knowing what each color is associated with the cones is more important. However, since we are focusing on the red-green on pigmentation, we would only focus the genes OPN1LM, or red ospin, which affects red light sensitivity, and OPN1MW, or green ospin, which affects green light sensitivity. During recombination, these genes, the OPN1LW and the OPN1MW are easily switched around, which leads to an absence of L and M cones. Colorblind can also be affected by the addition of abnormal O-spin in the pigmentation, but that doesn't really happen often. So the image below shows the OPN1LW and the OPN1MW causing color blindness, but it doesn't really explain how it affects color blindness, so that's what the next slide is going to go over. So now let's go over into more the molecular side of red-green color blindness. So going back to the previous slide, OPN1LW and OPN1MW can be switched around through a process called unequal crossing over. Look at the figure below at Part A. Part A describes the typical gene sequence of a red O-spin, green O-spin, and another green O-spin. The genes are X-linked and are adjacent to each other, resulting in the figure below. The genes are also 96% identical in the DNA sequence, which means during recombination, there's usually one red O-spin gene with around 1 to 4 green O-spin genes. We'll go more into why that is important in a bit later. So to explain red-green color blindness, it happens when the X chromosome crosses over. Because the DNA sequences are already so high, the gene sequences can misalign. During this, crossing over can occur, and if it does occur, then the recombinant chromosomes are not the same, leading to unequal crossing over. This in turn causes two outcomes to happen. One leading to the deletion of one red gene or the green gene, or a hybrid between the red and green gene. That is why there's typically 4 types of red and green color blindness, as it depends whether or not it's a hybrid gene or no red or no green pigmentation. This is where the ratio to red to green O-spin genes is important, because that means that it's not just a 25% for each type of red-green color blindness. 25% meaning that how likely they are to have it. Any one of those genes can be combined or deleted, but the fact that there's typically one red O-spin gene for every 1 to 4 green O-spin gene means that the ratios are different, and that's why out of the 4 red to green color blindness, deaneupanomaly is typically the most common one out of the color blindness, and that explains color blindness, red-green color blindness in general.
So today we talked about how red-green type of color blindness is the most common, especially in males, with about 95% of colorblind individuals being men. Color blindness is an X-linked recessive trait that is passed down through generations, so for an individual to be affected by the gene and express color blindness, the females must have 2 copies of each chromosome, and males only need it on the 1 chromosome, which is why color blindness is more common in males, causing it to affect about 1 in 12 men and affect about 1 in 200 women. We also go over how there's a 50% chance that daughters will have the gene for color blindness and pass it on, or will not pass the gene on to the next generation. And in sons, there is a 50% chance that they will or will not inherit the gene and express the trait. Next, we talk about how the different cones and rods found in the retina of the eyes, like the L, M, and S cones, affect high light levels. The rods affect low light levels, and how they affect the opsin pigmentation. Lastly, we go over how the OPN1LW and OPN1MW gene cause the red-green color blindness defects, like the absence of L or M cones, the production of abnormal opsin pigments leading to the destruction of the S cones or the defectiveness of these cones, and how these mutations affect red-green color blindness vision. And that concludes our video for today. Thanks for watching.